Welcome dear viewers to the informative video on dissolution method development for drug products with highly soluble drugs. So the current video is regarding dissolution method development, specification, standard conditions for dissolution of the immediate release formulations which contain the highly soluble drug substances. The highly soluble drug substances mainly are BCS class 1 and the BCS class 3 drug substances. So in this video we are going to have the understanding about the BCS classification in short, then dissolution requirements, disintegration and dissolution, method development, standard dissolution conditions, dissolution specifications, dissolution discriminatory power and regulatory queries. So I have referred the literature available for the dissolution method development. I have referred USP dissolution testing and EMEA reflection paper on the dissolution specification for generic oral immediate release products with systemic action. And along with this, I have put my experience onto the IR formulations containing BCS class 1 and 3 drugs. Also, I have put my experience onto the regulatory query handling for the immediate release formulations. So let's begin with the presentation now. BCS classifications classifies the drug substances into 4 classes on the basis of solubility and the permeability. But as we know, permeability is not in our hand and it is a basic and intrinsic property of the drug molecule. Permeability cannot be altered by the formulation, but sometimes the permeability enhancer and other technologies might be used, but for IR formulations mainly the solubility is critical. BCS class 1 are with highly soluble category and BCS class 3 are high soluble with low permeability. BCS 1 is with high solubility and high permeability and BCS 3 is with high solubility and low permeability. Then high solubility means the highest drug product strength and the highest single time dose or the highest single time dose is soluble in 250 ml or less of the aqueous media over the pH range of 1 to 6.8. Somewhere you will get the information like 1.2 to 6.8. So more or less these definitions are same. Also consider here that highest dose of the formulation and the highest single time dose. So don't confuse between these two terminologies. The first terminology of highest strength is if your product is having two strength like 100 mg and 50 mg, then 100 mg of the drug substance should be soluble. And if your drug product strengths are 100 mg and 50 mg, but the highest dose is 2 capsules or 2 tablets of 100 mg or 4 capsules or 4 tablets of 50 mg at a time or as a single dose, then it will become 200 mg. Then the main criteria for this dissolution specification and method development is the IR formulation, that is immediate release formulation, which are required to be swallowed. That means which are taken with water or which are swallowed and which are not required to be put into the cavities like sublingual root or buccal cavity or the rectal cavity. For those, these guidelines are not there. Also understand that this video is dealing only with the highly soluble drug substances and not with the low soluble type of formulations. For that I have made a separate video. You can get more understanding through that video. Then the main thing is The dissolution requirement are mainly it should be robust, it should be transferable, it should be specific to the product, then it should be specific to the formulation design. Like if you are working on tablet or IR formulation of tablet, or you are working on capsule, so if cross-linking is there in the capsule, that time pepsin can be used. So if the same molecule is formulated into tablet and capsule, that time you should try to make separate dissolution method and specification for both these type of formulation, still or though the molecule is same. Then it should be discriminatory, and it should detect the batches which may not be bioequivalent. So this is the main requirement of the dissolution testing. Now come to the disintegration and dissolution. So disintegration is breaking down of the formulation like capsule or tablet into the particles, and dissolution is the actual solubilization of the drug in the media. Disintegration test of the IR formulation are given into the different pharmacopoeias, and they have the limits like uncoated tablet, the disintegration time is 15 minutes, not more than 15 minutes. Film coated, not more than 30 minutes. Hard gelatin capsules, not more than 15 minutes. And if there are coating onto the formulation, that time some different disintegration time can be given, and if it is approved by the regulatory authorities. Our gastric emptying time is around 15 to 60 minutes during the fasting state. And also these DT and the gastric emptying time and dissolution are related to each other. The actual DT time of tablet or capsule, for example, is for some seconds to around 15 minutes or above it, but it is not much like 30 minutes. So you might have not came around the tablet formulation which is showing a DT of around 45 minutes or 1 hour. And if it is there, then you have to give the 2-time dissolution specification. 2 time points should be there, but it is very rare. Generally the 60-minute dissolution is the complete time for dissolution testing, and always the disintegration time versus the dissolution Q point should be correlated. It is very scientific. Then the method development. If you are working on to the formulations with BCS class 1 or 3 molecule, then the following points are very important. Selection of dissolution conditions. First you have to select the apparatus. It may be basket or paddle, that is USP 1, or paddle, that is USP 2. Then speed. It is 100 RPM for the basket, and generally IR capsules are tested by the basket apparatus. Then the USP apparatus 2, that is paddle apparatus, can be used with the speed of 50 RPM mainly as a standard condition, and 60 or 75 RPM if there is a coning issue. If there is a heap formation in the dissolution vessel, that time higher speed can be used, but it is required to be justified. Data of the RPM study should be submitted, and based on the regulatory authority approval, that can be used and finalized as a QC method. Media for the dissolution method should be in the physiological pH range, and surfactants not to be used, as these are not required for the formulations containing highly soluble drug substances. If your highest dose is soluble in the 250 ml or less media, and if you are using 500 ml media or 900 ml media, that time there is no issue of sink condition here. For highly soluble drug substances, sink condition is not an issue. That's why surfactant cannot be used, should not be used, or these are not required at all. Then coming to the specification and time point. So specification and the time point for the Q specification or Q point, that is finalized based on the bioequivalent or clinical study batch dissolution data, and based on that dissolution of the bioequivalence batch, the specification and time point should be given. The specification and the time point should not be too wide, like the formulation is rapidly dissolving or very rapidly dissolving in 15 minutes, and the applicant is giving a criteria of Q point of 80Q in 45 minutes, so that is not acceptable at all. Then standard dissolution conditions. For the formulations containing BCS class 1 and 3 drugs, the standard dissolution conditions are provided by the regulatory authorities like USA. It is also mentioned by the ICH guidelines and by the European agencies guidelines. So for standard dissolution conditions, the regulatory agencies are asking for mild dissolution conditions. And what do you mean by mild dissolution conditions? This means the speed of the apparatus, like paddle apparatus, should not be higher. 50 rpm for paddle and 100 rpm for basket with 500 ml media or 900 ml media can be considered as a standard and mild dissolution condition. Also, peak vessels should not be used. So for basket method, the stirring rate of 100 rpm or lesser rpm like 75 rpm may be used. For this is for some of the products. Then the media volume should be 500 ml or 900 ml, but generally try to select 500 ml because there is no issue of sink condition. Then the media should be 0.1 normal HCl in aqueous media or buffer with pH 4.5 or buffer with pH 6.8. So here the agencies always recommend dissolution in 500 ml media of 0.1 normal HCl, and the reason behind that is the disintegration time of the IR formulation is less, like 5 minutes to 10 minutes, and the drug substance is soluble. So within maximum 15 minutes, the drug product will release the total content of its drug substance, and that will happen mainly in the stomach. And in stomach, the acidic environment is there, and that is the logic behind selecting the 0.1 normal HCl as a dissolution media. Now, if you want to select other media like pH 4.5 or 6.8, that can also be accepted based on to the molecule behavior or drug substance behavior, like drug substance pK, drug substance solubility, drug substance stability in that media. The other medias can be justified. There should not be any surfactant and the temperature should be 37 degrees. For IR formulations, the dissolution is required to be run for 60 minutes and not longer than that. If the formulation is required to be run for above 60 minutes, that time it will not fall into the criteria or category of IR formulation containing soluble drugs. Because for some of the formulations, the solubility enhancement is done, though they are the IR formulations, they have longer DT, and 2-point specification is required to be given for such type of the formulations. I have made a separate video on 2-point dissolution. You can go through that to understand this terminology in a deep way. Then coming to the dissolution standard condition using paddle method or paddle apparatus, USP2 apparatus. The stirring rate is 50 rpm and higher rpm like 60, 75 for some of the formulations can be justified, and the basis of justifications for higher rpm will be coning or heap formation in the dissolution vessel. If there is variability in the dissolution results at 15 minutes and 30 minutes due to coning, that time you can justify the higher rpm. But along with the higher rpm, low rpm data is required. Dissolution medium will be 500 ml or 900 ml. The media will be 0.1 normal HCl or buffers with pH 4.5 or 6.8. Surfactant should not be there. But enzymes can be used in case there is a cross-linking in gelatin coated tablet or hard gelatin capsules. Temperature is same 37 degrees Celsius. The vessels are to be normal. Peak vessels or apex vessels should not be used. Generally, the peak vessels are not approved by the regulatory agencies. Lot of data is required to be generated, and generally the peak vessels are not required for drug products containing highly soluble drug substances. Then specifications. So how these specifications should be there and how these are derived. So specifications and the acceptance criteria are required to be the product specific. These specifications should be product specific and discriminatory in nature. These are based on the bioequivalence batch and the stability data. So whenever you are going to fix the dissolution specifications for finalizing the method, that time you should have the stability data. If you are going to take the validation batches, process qualification batches, scale up batches, that time you can give the relaxed specification and later on you can tighten those limits. And always remember, whenever you are going for validation batches, then you should give the 3-4 time points for data generation, like 15 minutes, 30 minutes, 45 minutes and 60 minutes, so that you will have data whenever there is requirement of tightening the specification, you can do that. So for example, here I have taken hypothetical examples to understand about the specification. So consider you have IR formulation or IR tablet or capsule formulation with 60 minutes as a dissolution runtime. And for example 1, this is the data for dissolution. This is mean data. You can consider the data for 12 units. Here in 5 minutes it is 78, in 10 minutes it is 95% release or dissolved, and in 15 minutes it is giving 100%. Now for this example you have to give the specification of not less than 80% Q. If for example 1 you give limit of 80 Q at 30 minutes, then it will not be accepted by the regulatory agencies. Similarly for example 2, you can give criteria of not less than 80 Q at 30 minutes. And just an example I have taken as example 3, which is giving the dissolution of above 85% in 45 minutes. So this is the requirement that the specification Q point should be 80% Q at 45 minutes. But always remember, generally the specification for IR formulations containing BCS class 1 and 3 drugs, 15 minutes or 30 minutes is sufficient. And if it is showing slower release, then you should improve the RPM or speed of the paddle apparatus, or you should take other appropriate action. But always try to give the specification of ATQ at 15 minutes or at 30 minutes. Then discriminatory power. So I got some comments for proving the discriminatory power of formulations containing highly soluble drug substances. And please understand here, the answer for those comments and questions is given in this slide. Whenever you are working onto these formulations, you have to have complete data for studying the impact of CMAs, CPPs, process and storage conditions onto the dissolution. You have to understand about the criticality of the dissolution specification versus the critical variables which have impact onto the dissolution, like critical material attributes, which may be the excipient grades, excipient particle size, excipient LOD, that is loss on drive, particle size of the API, polymorphic form of the API, and many more. Then critical process parameters. Then process and storage conditions. So as I have mentioned earlier, stability data should be evaluated before finalizing the dissolution. So stability is very important. Then the efforts to get discrimination should be provided with dissolution data. For these type of formulations, generally the discrimination cannot be achieved, and generally all the batches with different CPPs, CMAs and storage conditions will give you the rapid dissolution. But still the regulatory agencies wants to understand the product behavior in dissolution. That's why the regulatory agencies ask for the data or want to see the applicant's efforts to get the discrimination. After all, if there is little discrimination or if there is no discrimination, still it is okay. But one thing here is to remember is that your dissolution conditions and the dissolution specifications should be standard. If you are giving too wide specifications and then you are trying to say that I am not getting discrimination, that is not at all acceptable. You use the mild conditions, you use the standard conditions of dissolution, and then try to get the discriminatory power. So the efforts to get the discrimination to be provided in the application. Discrimination is difficult for drug products containing highly soluble drugs. The dissolution condition should be standard and mild conditions should be used. Specification is required to be based onto the BE batch dissolution. Specification time point and Q time point should not be too wide. So if this criteria is met, then the queries will not be there for discriminatory power. Now coming to the regulatory queries. I know the video is getting little bit lengthy, but have patience, you will get the complete understanding about this topic. So what are the regulatory queries? One of the most famous, I can say it is a famous query, that tighten the dissolution specification. So tightening the limits is always a famous query in the formulations. Dissolution specifications based on the dissolution result of the batch used in the bio studies or clinical studies. Time point like 30 minutes to 15 minutes. If you have given a time point of 30 minutes, the agency will ask you to make it 15 minutes. And the data should be there to support that specification to be tighter. Then percent dissolved to be 80% Q. If you are given 70Q at 30 minutes, and if you are getting good dissolution at 15 minutes, then the regulatory agency will ask to make it 80Q at 15 minutes. Then what are the critical attributes for dissolution? So agency asks for the critical attributes for dissolution. How the media and other dissolution parameters were selected? This is one of the very frequent question. How you have selected the volume, how you have selected the media, and how you have selected the RPM. So these things are required to be the part of the dissolution method development. For most of the formulations, USP apparatus 2 is used, and as per my experience and understanding, mainly for IR formulations, USP2 apparatus is used, and if capsule formulation is there, then basket apparatus can be used. Though the applicant used monograph method, still dissolution method development and discriminatory power studies are required. This is very important statement here. You cannot justify or you cannot say in your development report that you have used a monograph method and that's why you are not going to make the dissolution method development report, or you are not justifying the discrimination. That is not accepted. Whatever the method you are using, that method is required to have development report and discriminatory power in the report. Then along with that, solubility studies are required, and generally the drug substances shows high solubility in all the medias, and as the BCS class 1 and 3 are given on the basis of highest dose solubility in 250 ml media, and if you are going to use 500 ml, then sink condition problem is not there. So these things are interrelated, and dissolution is a very vast topic. Dissolution is very important performance test for the oral formulations, and dissolution is the topic of great interest for the interviewers, and if you have good understanding about the dissolution, then there are great chances for your selection. And generally for dissolution, the interviewers are going to ask you 3-4 questions, and based on those questions only they can decide to select you or not. So keep learning, keep watching Pharma Learning in Depth channel and have great understanding. Also, I will be happy if you give your comments, suggestions for making the videos on the topics which you are looking for. Thank you for watching the video. Let's meet you soon with such kind of informative video. Till then, take care and bye.